Well, good afternoon, everyone, and welcome to the third webinar in the Animal Science Research Webinar Series for 2019. I'm Chris Hosteller. I'm the Director of Animal Science for the National Pork Board. I'm excited to introduce today's speaker, but first I've got a few housekeeping items. As a reminder, these webinars are designed to highlight research results from completed projects that were funded through checkoff dollars. This is a vision of the Animal Science Committee as they thought about ways to disseminate the results of completed research projects that were supported through this mechanism. So this year is actually our 6th year for these webinars. They're held each Tuesday in the month of August at noon Central. You can view the previous 20 webinars by visiting Animal Science page at pork.org. If you haven't done that already, I would encourage you to do so. There's a lot of good information in those previous webinars. Also remind you, at the end of Dr. Gabler's presentation today, we'll have time for question and answers. Please type those questions that you might have into the chat box, which is located in the lower right-hand corner of your screen. Be sure to direct those to me, and then I will relay those questions to Nick for his response. You don't need to wait until the end of the webinar to ask those. Just feel free to write those to me as they occur to you. Today's webinar is presented by Dr. Nick Gabler. Dr. Gabler obtained his bachelor's and PhD degrees from La Trobe University in Melbourne, Australia. Upon completion of his PhD, he conducted a postdoctoral research study at Purdue University and then at Iowa State University. In 2008, he joined the Animal Science Department at Iowa State University as an assistant professor. He's now an associate professor in that department. His research has been focused on fundamental swine nutrition, metabolism, and physiology. Dr. Gabler has an active and diverse research program that focuses on understanding and improving swine metabolism, growth, and feed efficiency. And over the past 8 years or so, Nick has conducted a series of checkoff-funded research projects, really digging into the interface between swine health and nutrition. The title of today's talk by Dr. Gabler is Nutrition of Health-Challenged Pigs. Health challenges are certainly one of the major causes of pig mortality. How we feed pigs leading up to, during, and following a health challenge has substantial impact on their recovery and subsequent productivity. Dr. Gabler will discuss nutritional approaches to help sick pigs through these health challenges and how that can translate into ultimately into reduced pig mortality. Also, within a day or so of this webinar, we will post the link to the recording of it on the Animal Science page within pork.org. And if you go there, you'll notice that we've got last week's posted there as well. So you'll be able to see a summary of that and then also listen to the recording. So with that, I'll turn the presentation over to Dr. Gabler. Nick?
Okay. Thank you, Chris, for that introduction, and good afternoon, everyone. So today I'm going to give, Chris asked me to give an overview of some of the research that we've been doing in collaboration with the Pork Checkoff, and I really appreciate their funding support over the last 8 years for some of this work, and also some of the work that ties into that we've been able to piggyback on to actually obtain some USDA NEFA funding for. And so this work really is really we've been trying to understand the best way to feed and manage health-challenged pigs. And so to actually do that, we've been focusing primarily on nursery to finisher pig pathogens. And then the work that I'll present here today combines pathogens such as PRRS virus, PEDV, Lawsonia intracellularis or ileitis, some Mycoplasma pneumoniae challenges. I'll present one data set on some swine influenza. And then we're just finishing up a study with brachyspire, or swine dysentery, in which we've been looking at the interaction with dietary fiber. And so a lot of the actual work that we've been conducting over the last probably few years has been trying to understand how do these pathogens, in particular at the appropriate age of the pig, how do they actually alter nutrient partitioning and metabolism of the pig? And then are they having a big impact on pre-absorptive metabolism or post-absorptive metabolism? In other words, if these pigs are going through a health challenge, is there an opportunity to actually do something to improve either digestibility and nutrient uptake, or can we focus on energy and nutrient partitioning? And pretty much in a nutshell, we're seeing from this research where we've evaluated lots of different diet combinations with different pathogen combinations, it falls into two categories. One category is that we can do some interventional strategies that can actually improve survivability, but then has no impact on pig performance. And the other kind of part of that is that we can do some stuff through a diet management that can actually improve growth rates, but then doesn't really move the needle or actually do much with survivability per se. So when we think about nutritional interventions or nutritional strategies, we can probably think about one, to just improve survivability of a nursery to finish a pig, or the other one, are we actually improving growth performance and feed efficiency, etc. We haven't got any silver bullets because I think it depends on the severity of the pathogen burden itself. So what is the pathogen? Is it bacterial or viral? Is it clinical, subclinical infection? Is it multifactorial in nature? And then that would all kind of depend on what type of nutritional strategy may be best. But there's no silver bullet saying that diet X or Y or Z is actually going to be more beneficial to actually improve growth performance or survivability. But I'll share some of the data that we've conducted over the last couple of years on that. So one of the problems that we have with disease challenges is that there's a metabolic priority for resource allocation in the pig. So in other words, there's a high metabolic priority towards immune function, towards brain function, placental fetal development, etc., and then a lower metabolic priority towards visceral organs, bone, muscle, and adipose tissue mass. So when a pig is actually health compromised, muscle and adipose tissue accretion is actually compromised. And so therefore, what can we do to actually mitigate this? And part of it is driven by pro-inflammatory cytokine and hormone-driven responses. And so anabolic and catabolic signals are actually improving, or say diverting or re-diverting resources away from lean tissue accretion more towards more towards the immune function. And so our disease hypothesis that we've been testing for the last probably 8 years can be summed up with this cartoon. We typically see reduced nutrient intake in a pig that is health challenged or health compromised. Let's see if my cartoon works. Here we go. Let's go. Sorry. The cartoon's not my cartoon is not going to work, I don't think. Oh, here we go. And so we have reduced amino acid and carbohydrate and energy intake in these pigs that are health compromised. And then we've tested the hypothesis, does this disease burden actually increase free radical production or reactive oxygen species and therefore increase muscle proteolysis to liberate amino acids that then go from lean tissue accretion towards the liver and the immune function. So we know there's an increased need for energy, there's increased need for amino acids and other protein sources for the febrile response, in other words, the fever response, and also to build antibodies, build cytokines, acute phase proteins, etc. And so does this actually compromise lean tissue accretion, or to what extent does this compromise? And then there's increased need for gluconeogenesis and glucose into circulation. There's a lot of work in the biomedical literature to suggest that glucose and glutamine are important metabolic triggers to help with T cell and B cell metabolism as well as macrophage metabolism during a disease challenge. So how does this all lead to a compromised lean tissue accretion in these pigs? And so we've been testing these hypotheses, and some of these we're actually going to reject, and I'll touch on that at the end. So really, when we talk about PRRS, this is just a sole PRRS challenge, so individual PRRS challenge that we've published. This was one of our first pork board projects that we conducted. And really what we're interested in was a longitudinal impact of PRRS. And so this is just really the first 42 days. These pigs started off at about 70 pounds, and and we went up to about 150 pounds. But we could see over this challenge period that the control pigs has outperformed the PRRS-infected pigs, where we had about anywhere from about a 15 to 20% increase in performance in our healthy pigs over our PRRS pigs. Or in other words, we had about a 20% decrease in pig growth rates, feed intake, feed efficiency, and also in tissue accretion, which I haven't shown here. And then if we took these pigs all the way out to market, so we take them out for about another 100, to about 110, 114 days post-infection, these pigs were actually leaner and they had reduced yields when we took them to the slaughter plant. And so really what we looked at with these pigs, if we graph our kind of growth or body weight gains over time, our black line here is our control pigs that were PRRS naive, and then our red line here is our PRRS challenge pigs. It took our PRRS challenge pigs about 14 extra days to get to the same body weight. So the same market weight are about 127 kilograms. And then we and others, so Schwer was the PhD student working, Wes Schwer was a PhD student working on this work, and others have reported that if we do a napkin math, we estimated that this PRRS infection model in this set of pigs, and these were maternal line barrows, cost anywhere between $3 to $30 a pig, and that would of extra cost that would be associated with the cost of the PRRS infection only. And we had no mortality or secondary infections associated with this. So this cost would be higher if we had mortality associated with it. But in a nutshell, a PRRS infection during the growing period had a long-term impact on the performance of these finishing pigs. We see the similar thing with a dual challenge that we conducted with a Lawsonia and Mycoplasma hyamoniae challenge study. In this case, we inoculated about, these would be about 130-pound pigs, 60-kilogram pigs, and we gave them pretty much it was a subclinical challenge of Lawsonia mycoplasma. And so we had mild lesions, we had mild lung lesions, I should say, and also small intestinal lesions, and also some mild clinical signs of this disease. But even with a subclinical mild challenge here, we still resulted in about a 17% reduction in average daily gain over this 42-day period, test period, challenge period, and about 11% reduction in feed intake over that period of time. And that translated to, if we look at it on a tissue accretion basis, so whole body tissue accretion, we still end up with a similar, about a 15% reduction in lean accretion and protein accretion in these pigs, and fat accretion due to the dual challenge. Some other work that we did in collaboration with NutriQuest and Phil Geiger, they gave us a data set to look at because we were interested in what was the impact of a swine influenza challenge in a nursery pig. So these pigs started off at around 7 kilograms, and they went for 28 days post-infection, and then they ended up at about 22 to 24 kilograms at the end of that study. But really what was interesting here with influenza challenge in a nursery pig, during the first 5 to 14 days, we see a reduction in growth rates or body weight gain in these pigs. And that translates overall. And then really, but once they get past 14 days, actually the growth rates is not shown here, actually increase. So there is some compensatory gain with influenza once they get past that kind of 14-day period. But overall, there was a reduction over this 28 days in average daily gain in these pigs by about 7%, but feed intake actually increased over this 28 days. And this feed intake increase, it was really driven by the post-14-day DPI feed intake response. In the first 5 days, the actual feed intake was decreased significantly in these pigs, but then after they've actually kind of started to resolve the virus, the flu challenge, then feed intake went up, and that was actually part of their compensatory mechanism. And so really it comes into where you want to sample, but take your data points, you can have different impacts. But the thing here is that these nursery pigs, even though they were challenged, they were compensating once we got past 14 days in this challenge. Back in 2015, we started doing some work with porcine epidemic diarrhea virus as well, and this is work from Dr. Shelby Curry. And then really with porcine epidemic diarrhea virus in nursery pigs, what we see is like we see in suckling pigs, we see that we get that kind of thin-walled intestines at about 5 days post-infection compared to the control pigs up here in this top left panel. And at 7 days, these intestines, this bottom right panel, are starting to look more normal. So in a nursery pig, unlike a suckling pig, these pigs do kind of turn over their intestinal epithelium relatively fast, and actually they do recover. And then how that translates into performance parameters, really we see again about a 16% reduction in average daily gain over a 42-day challenge period, and then a 27% reduction in feed intake over the same period. And unlike what, a suckling pig, these pigs do they're shedding the virus for about 7 to 10 days, and then by about 14 to 21 days, that virus, the PD virus, seems to be kind of resolving itself, and the intestines are actually looking more normal. But there is a lag effect with regard to tissue accretion and growth performance in these pigs. And so one of the opportunities that I think we have with actual, at least some enteric pathogen challenge such as PED, is actually looking at ways to actually improve recovery in these pigs. We've tried a few things looking at some water-soluble nutrients deliveries to PED-infected pigs, and we didn't see any benefit from a performance parameter aspect in those pigs. But I think if in this study here, this is really just showing intestinal changes. So we see over time in these white bars here, there's a decrease in villus height, and also crypt depth is changing. But then by day 14, villus height is back up to the same level of control. And really that's indicative of actually stem cell proliferation in the base of the crypts. So Ki67 is a marker of stem cell proliferation in the crypts of pigs. And so you can see down here in our control pigs, all these red dots down here are positive stem cells that are proliferating, regenerating. But you can see at day 5, there's a lot more proliferation occurring in the PDV-infected nursery pig than what there is in the control pig. In other words, in the first kind of 24 to 48 hours, you see that significant blunting, attenuation of the intestinal epithelium, so villus height is shrinking, crypt depth is changing, getting very thin-walled, but by day 5 to day 7, these pigs are actually starting to increase cell proliferation and got a lot of immature and absorptive cells that are actually proliferating, and then they'll start to migrate up the actual villus and start to then regenerate this epithelium. One thing we actually looked at with this is that the nutrient supply to rebuild this epithelium does not appear to be coming from the liver or from the muscle of the pig. It actually seems to be more luminally driven nutrient supply. So Dr. Curry has published some of this work when she was a PhD student with me now, but then the data would seem to suggest that if we want to help these pigs recover, we need to give them a dietary or oral kind of nutrient supply. So one thing that someone could do is actually probably give pigs more branched-chain amino acids, such as leucine, could be a way to actually improve the stem cell proliferation and actually give the amino acids and or give the amino acids and the protein that's needed to rebuild this epithelium to help these pigs get back on their growth curve better. So something to do more with diet, and this is some work that's literally just finished about 2 weeks ago, the actual live animal part of it, is that we're interested in the impact of how swine dysentery or brachyspira challenge interacts with dietary fiber. And so some work in 2014 by Dr. Eric Burrow at the Iowa State Vet School, and his PhD student at the time, Dr. Wilberts now, showed that feeding a 30 to 40% corn distillers grain diet actually increased or augmented swine dysentery disease in growing pigs. And so what we're interested in doing, based off some of the European and Australian work, is can we change that fiber profile to actually mitigate swine dysentery in these pigs and actually improve pig performance? So what we ended up doing for this study is that we had 3 treatments. We had these pigs start off at about 70 pounds, 35 kilograms, you can see here. We had a negative control that was fed a 20% corn distillers diet. We had a positive control that was fed the same 20% corn distillers diet, but was challenged with swine dysentery or brachyspire. And then we had a positive challenged group of pigs that were fed 10% distillers grains, 5% resistant potato starch, and 5% sugar beet pulp. So we're trying to put in a more fermentable or soluble fiber source in place of 10% distillers grains. So they still had 20% kind of fiber, or at least 20% inclusion rate of these products in the diet. And then what we did is that we euthanized these pigs in a window of 10 to 16 days based on each pig showing about 3 consecutive days of clinical infection. And so I haven't got all the data here with me right now, but really what we did is a lot of the positive control pigs actually were euthanized in about day 10 to 13, and then from day 13 to 16, most of the resistant starch pigs started to become positive. And so really what we're seeing here is that our changing the fiber source, or decreasing the corn distillers and increasing the soluble fiber, actually improved growth rates over this challenge period. And then also improved feed intake compared to our positive control pigs. And so by changing the fiber source, we can actually, in this case, increasing resistant starch and beet pulp, seem to delay the peak disease and improve performance over the peak disease period. Now, one thing that we don't know, and even the published literature hasn't really done a lot of work in this, is it a delay? In other words, if we gave these pigs another week, especially the resistant starch pigs, would they actually become worse, or were they actually at the point of resolution when we actually euthanize them for tissue collection? And so that's a follow-up study that we need to do. Is changing the fiber source just delaying the onset of disease, or is it actually decreasing the severity of disease? But there's an opportunity to play around with at least with distillers grains and soluble and insoluble fiber in these diets. Some similar work has been done. I know John Patience here at Iowa State has done some work similar with E. coli challenge work, showing that beet pulp, 10% beet pulp in nursery pigs is beneficial for E. coli challenges. In other words, it decreases the incidence and shedding profile. And this is, I think, an area that is worthwhile further pursuing, is how does a fiber source or composition in the diet actually mitigate bacterial disease? So a lot of these studies that have quickly gone through is really showing that disease has an impact on growth and also intestinal function, but to what extent? And does viral disease have the same impact as a bacterial disease? And then feed intake seems to be a common denominator that seems to change. And one of the first signs of a problem or disease problem in a barn is actually you'll see the pigs go off feed intake. So what we want to know is, to what extent does feed intake explain the disease phenotype? And to do that, we did a USDA-funded study looking at porcine reproductive and respiratory syndrome virus. And so this study we conducted last year, but really what it is we had 3 treatments. We had a 10-kilogram nursery pig is what we used for this. We had pigs that were allocated to either PRRS challenge group, which would be here in red. We had pigs that were allocated to an ad libitum fed, allocated to a non-PRRS challenge group, and that would be black here. And then we had another group of pigs that we actually fed that were non-challenged with PRRS, but were fed the same caloric intake one day behind our PRRS challenge pigs. So that's called our pair-fed or restrict-fed group. So what we want to know is that if the pigs were getting the same caloric intake as a disease challenge pig but didn't have the disease or didn't have the PRRS virus in them, what was the phenotypic clinical signs of that pig? And so what we end up doing is, if you look down the bottom left panel here, is that 1 day behind we fed the pair-fed pig, or the restrict-fed pig. So in this case, if our PRRS-fed pig ate about 400 grams of feed a day here, the next day the actual pair-fed pig got 400 grams. And then if it went down to 200 grams, then the pair-fed pig the next day got 200 grams. So we tried to calorically match these pigs all the way through for 35 days. And so what we can see here is that our ad lib control pigs are here in black. They had a nice increase in feed intake. They peaked up at about 1.4 kilograms a day. And then during our peak disease, these pigs, we went down to about 200 grams a day. And so we had a huge difference in feed intake in our pigs. Just to show here that our control pigs, both our ad lib and strict or pair-fed pigs, remained PCR negative for PRRS throughout the 35-day study, where our PRRS-infected pigs by day 4 had circulating virus in their serum based on the PCR, and then they're starting to resolve that, but they're still positive of shedding virus at 35 days. And then on the right-hand panel over here, we just show the antibody response. So the antibody negative for our ad lib and restrict-fed pigs, and our PRRS-fed pigs were starting to seroconvert by about day 7, and then they were peaking by about day 14 to 21 with regard to antibody production. The other thing that we did with these three treatments, that we microchipped each pig to look at daily body temperature. And we can see here that PRRS in our red here versus our ad lib control pigs in black, PRRS initiated a febrile response starting at about day 5 and ending at about day 15 to 18. And so there was a sustained increase in body temperature in these pigs. And you don't get this type of response using an endotoxin or LPS challenge. Normally you'll just have up for 6 to 12 hours and back down, up 6 to 12 hours and then back down with regard to endotoxin challenges. So the PRRS initiated a nice sustained febrile response, but our pair-fed or restrict-fed pigs actually had a decrease in body temperature compared to even our ad lib control pigs, and that was due to the fact that they were just fed less, and the thermal effect of eating actually just reduced their core temperature. So between these two treatment groups, talking about 2 degrees difference at peak kind of viremia seroconversion, peak disease here. So what does all this mean for the overall 35-day performance in these pigs? With this model, and this was a 134 open reading frame 5 PRRS isolate we used for this particular study. Here we see that PRRS reduced feed intake by 51%, and our pair-feds were identical by design, you see here, compared to our ad lib control healthy pigs. And then average daily gain was reduced by about 60%, 58%. And it was very similar for our pair-fed group. And then feed efficiency was not different between treatments. So this is automatically showing us that actually caloric restriction during a disease challenge or disease hyperphagia or disease anorexia, whatever word you like to use, that the disease pressure on appetite seems to explain at least a lot of the growth performance phenotype we see with PRRS in our model. And so how did that translate into body accretion? Really across the board, we see a 30 to 60% reduction in fat accretion in grams per day, protein or lean accretion, depending on which unit you want to use, and total mass here when we look at tissue accretion in these pigs. The other thing that was different in these pigs with regard to this disease model is that our PRRS pigs were 15 kilograms lighter in body weight compared to our ad lib pig. So here's our ad lib pig here on the right. If we look at this bottom picture, on the bottom right picture here, here's our ad lib fed pig, here's our PRRS pig in the middle, and here's our pair fed pig. You can just see these pigs all start off at 10 kilograms body weight, and just over 35 days, there's already 15 kilograms difference. There's 15 kilograms lighter. And our pair-fed pig here is quite similar in about 15 kilograms. But they had larger intestinal mass compared to our PRRS pigs, where our PRRS pigs had larger mass with regard to lung, liver, and heart due to the actual consolidation of the lungs and the pluck in these pigs due to the disease. So they are repetitioning energy differently. If we want to look at the loins in these pigs, here's just an example of an ad-lib fed pig. If you look at this center left image, here's the loins in these pigs. Here's an ad-lib pair-fed and PRRS-fed pig. We took the loins out of these pigs here. And this is at day 16, 17, where we virtually had minimal growth rate or no growth rate, negative growth rate actually on these pigs. And we also had a little bit of growth rate on our pair-fed pig. But really what we're seeing here is that feed intake seems to describe a lot of this phenotype. The other thing that we've done, and I haven't presented it here, is that we've actually went in and looked at muscle proteolysis and protein synthesis pathways in these 3 loin tissues. And then what we see is that it's really just a nutrient supply issue. In other words, it's protein synthesis through the mammalian targeted rapamycin pathway is just inhibited. In other words, there's not enough energy or amino acid supply to stimulate muscle growth or protein synthesis in these muscles compared to what we see with our ad-lib fed pig. Proteolysis pathways, if we look at the proteasome, ubiquitin proteasome pathway, we look at the caspase pathway, we look at kelpane, kelpstatin, we don't see a lot of difference between these 3 treatments. And so these pigs are not necessarily mobilizing amino acids to support growth at all. They actually don't seem to be doing that. They seem to be preserving. And then we're really seeing it's a nutrient supply issue rather than actually a mobilization that's leading to differences in growth rates and lean accretion in these pigs. So if we put this on a energy expenditure basis, what we end up doing just on some napkin math, that these pigs were consuming about 4,400, 500 kcals of ME intake per day in our ad-lib pigs compared to half of that. So we're up around probably about 2,000, 2,200. And we can see across the board here, I'll put it on a percentage basis, is that the PRRS and pair-fed pigs are petitioning less energy towards lipid deposition and also protein deposition compared to the ad-lib fed pig. And then there's a lot more energy in our PRRS-fed pig going to non-lipid or protein deposition pathways, and even in our pair-fed pigs. So there is some mobilization of how these pigs are utilizing it. And if we do the math here, we've calculated it's about 150 kcals per day of ME intake differs due to the virus. And that's mainly due to the febrile response and the immune response associated with these pigs that we see with PRRS. And so there's a deficit there that cannot be explained by just caloric intake. So really, appetite explains most of the PRRS disease phenotype, and I think we see the same thing if we're talking about Lawsonia, we talk about swine dysentery, we talk about Mycoplasma hypomoniae through feed intake. So really, what can be done about this? And this is probably a bit where the nutritional strategies may come into it. So I'm going to just quickly mention 2 projects that the Pork Board funded us and Iowa Pork Producers funded us. The first project, this 15099 project, is where we looked at trying to deliver nutrients and energy through the water and also through a top dress. In other words, with this study, we used a kind of like a Blue 2 liquitine type hybrid product that was formulated. We put it through the water lines or we top dressed it. So this product was a combination of proteins and amino acids so soy protein isolates. It was a combination of, had some monosodium glutamate in it, some high fructose corn syrup, some electrolytes, and even some vitamins and minerals in it. And then we gave this supplement either through the water or top dressed in the feed for about 6 weeks in our PRRS challenge model. And really, at the end of the day, we did not see either of the top dress or the water supplements support peak performance, i.e. growth rates or feed intake in our PRRS infection model that we did in one of our earlier studies. It may have had an impact on survivability, but we didn't have enough numbers. We only had about 100 pigs on test for this study, but we did not support growth by providing stuff through the water lines, which was what we thought we would. We thought we'd be able to enhance growth rates. Water intake was good in these pigs, so water intake didn't seem to describe it. What we did see when we did provide kind of nutrients through the water lines, that actually pigs, they ate less out of the feeder. So there's some tweaking that needs to be done with kind of, at least we'll say, water formulations, trying to plumb it into the water line to support growth in pigs, because we don't see that. We didn't see any benefit here. Actually, it was more of a hindrance than anything else. Also, the addition of extra glucose through the feed or water did not exacerbate any secondary infections. Quite a few people have said that if you put extra glucose or extra carbohydrates in the feed during a health challenge, it's going to help proliferate pathogenic bacteria. We didn't have any problems with any of that. The other thing that we've tried to do in the past, and this was the 15148 project that the Pork Board funded us, was to evaluate soybean meal replacement in growth finished pigs when they were actually given the dual challenge of PRRS and mycoplasma. And so with this one here, Wes Shuir, when he was a master's student with me, actually we went in and we dual inoculated some about 200-pound pigs, and we gave them a PRRS and mycoplasma high ammonia dual challenge prior to market, so about 4 to 6 weeks prior to market. And then with that, we fed them either, it was about an 18% soybean meal diet for our high soybean meal, or we actually fed them about 8% soybean meal with some synthetics to meet their amino acid requirements in these late finishing pigs. So if we compare 8% to 18%, we actually did not see any benefit of feeding, we'll call it high soybean meal, or at least soybean meal, to these pigs during a late-breaking respiratory dual challenge. Now, we probably need to go higher with soybean meal, may have to go up to 30 to 40%, but that was just unrealistic from a practical standpoint in a 200-pound pig. But we didn't see any benefit of soybean meal replacement or over synthetics in late finishing when we tried that. Some other work that we've done, we've actually tried to manipulate appetite, and we did this study last year. What we tried to do is, can we manage the PRRS phenotype, especially the appetite phenotype and the febrile response to the fever, if we give these pigs either a vitamin B12 treatment, so we injected pigs with vitamin B12, and we also fed them at the same time. We gave them oral drench of sodium salicylate for 14 days. So they had every 7 days they had a vitamin B12 injection, and then we fed them sodium salicylate. So that would be the gray bars up here. So or we fed them tonicity, the tonicity product, to see if we could actually improve feed intake and actually have a benefit on growth. So really what it comes down to, our black line here is our control pigs that were just non-challenged for the control diet. Our grey bar here were pigs fed the actual a B12 supplement or injected with B12 and give them sodium salicylate for 14 days. We didn't see sodium salicylate or the B12 either improve appetite, back over here on the right panel, or actually decrease body temperature. And then the tonicity, we didn't see it actually do anything with febrile response or body temperature. However, it did kind of, we'll say, numerically do something the first couple of days with regard to feed intake, but thereafter it didn't do anything with regard to driving feed intake in these pigs. So even if you had something that's more highly palatable, it didn't drive feed intake, whether it was given as a top dress or as a liquid supplement here. But what we did see with this is that, the bottom left panel, is that having either the tonicity product or the sodium salicylate B12, having some sort of, we'll call it simple intervention strategy, did improve survivability in these pigs. So it was not doing anything in this red line. We end up with about 75% mortality. But then if by doing some sort of intervention, whether it's with a isotonic electrolyte supplement or with B12 sodium salicylate, here in the grey, we actually improve survivability in these pigs. but we didn't do anything. It didn't move feed intake. It didn't do anything with growth rates. So I'm an advocate. If we do have a severe, this was a pretty hot PRRS challenge model that we used. This is on low numbers. This needs to be repeated out in the field for larger numbers. But giving either some sort of intervention, whether it's sodium salicylate, B12 injections, or doing something a bit more practical, or giving them some sort of isotonic supplement, whether it's tonicity, blue light, blue 2, a liquidine type product, seems to have a benefit with survivability, improving survivability. They're not doing anything at all. So in the last kind of nutritional study that we've been conducted, or this is our is a set of studies that we've done with the pork board, is we want to know, can we actually change the lysine to energy requirement of the pig to actually optimize performance during health challenges? And so the data out there was pretty limited of what is the optimal ratio of lysine to energy in a pig. So Wes Schwer, as part of his PhD program in 2018, through this pork board funded project, we did a titration in 25 kilogram and 50 kilogram pigs in collaboration with the mash-offs, and we want to know what was the ideal lysine to energy ratio. And so he did some breakpoint analyses here. This paper is published in Translational Animal Science. You can get that reference, or I can provide it to anyone who wants it. But really what we see here is during a health challenge, or in healthy pigs or in health challenge pigs, in our health challenge pigs, if we move the lysine to energy ratio to be 110 to 120% of requirement, so if it's 100% is the ideal for a healthy pig, if we move it to the right 10 to 20%, we can actually improve growth rates and feed efficiency during a health challenge. They'll never get back to the same kind of rate of growth or feed efficiency as a non-challenge healthy pig, but in other words, for a 25 kilogram pig, we go up to about a 3.2 lysine to ME, grams of lysine, SID lysine to ME. 3.7 for an older pig seems to be ideal. And this seems to be beneficial if it's actually a natural or experimental challenge. And so, in other words, if the pigs naturally broke with PRRS or experimentally challenged with PRRS, we seem to see a benefit with regard to supporting growth in health in a PRRS challenged pig by moving that lysine to energy ratio by 110, 120% to the right, so increasing it. And so our current study that we're wrapping up with the pork board by the end of this year is we want to validate that. So we want to validate, can we actually improve growth rates and feed efficiency of a PRRS challenged pig if we have a lysine to energy ratio of about 120%. So in other words, so for this study, we set our grams of lysine. In this current study, we set it for about 2.69 grams of SID lysine per ME, which is 100% for this age pig that we used, so 35 kilogram pig. And then we did 20% on top of that, which will be a 3.23 grams of SID lysine per ME and a 3.23 grams of lysine, of which we achieved the same ratio by actually diluting energy instead of increasing. So we had a control diet here, the 100%. We had a 120% diet, which we just increased amino acids and protein by 120% or 20% increase over here and kept energy the same. Or we did the other diet, this third diet, we actually just diluted energy by about 20% and then tried to keep amino acids and protein the same. So in other words, we want to know, is the ratio more important than the actual amino acid or protein total content of the diet? And we did this in PRRS vaccinated and non-vaccinated or not vaccinated pigs. During this study, they actually they were not vaccinated for PCV2, so they actually broke with PCV2 at the same time we gave them the PRRS challenge. So we had a really good disease model that ran about 14% mortality, and that was really a combination of PRRS and PCV2 co-infection that we saw in these 35 kilogram pigs. One thing that people always ask me, okay, if we do something with the diet, are we actually altering the serology and virology of these pigs? And then really I'm just putting this up here just to show that, one, our vaccinated pigs here, which was with the BI Inglevac vaccine, actually they seroconverted a lot faster compared to our non-vaccinated pigs. So by somewhere between day 21 and 28, they stopped shedding virus and they're seroconverted, because they've already seroconverted prior to that, where it took about 14 days for our non-vaccinated pigs to seroconvert. But the other thing is that whether we changed the lysine to energy ratio of the diet, it didn't seem to have a major impact really on the serology or virology of the pig. So this type of diet did not alter the PRRS shedding profile. So we're trying to support growth. So did we, over this 42-day challenge, did we actually support growth? So here we're just comparing within the PRRS vaccinated group and non-vaccinated group. So we did the comparison separately. So these pigs are all vaccinated at weaning with the BI Inglevac PRRS vaccine, and we can see here that they're all challenged with actual PRRS as well at about 35 kilograms. And we can see here that average daily gain was significantly increased due to 120% ratio of the diet, and also the growth rate increased if we actually diluted that ratio, or got the same ratio but diluted it with sand. So in other words, if we dilute energy or increase the amino acids to achieve this ratio, we're actually supporting growth over our 100% pigs. And then a lot of this, I call it the sand diet, because we use sand to dilute it. With the sand diet, we're actually driving feed intake to get the same rate of growth. So feed efficiency is not good, but they're driving feed intake to actually eating more. So if we dilute energy in the diet, these pigs that are health challenged and vaccinated are increasing feed intake. In our non-vaccinated pigs, you can see the non-vaccinated pigs they were worse off. They were about 10 kilograms behind our vaccinated pigs, so it pays to vaccinate. That's the first story. But really, we see the same thing at 120% with amino acids driving that ratio. We see improved growth rates, and then we see the same thing if we dilute energy to achieve the same ratio. Driving feed intake, we get similar growth rates to 120%, and both of them are better than our 100%. So in other words, what we're really seeing here is that sick pigs will eat to their energy needs. If we increase that ratio, we validated that we increased the ratio 120% of lysine to ME. We kept all the other amino acid ratios to lysine the same. We can actually improve pig performance in a health-challenged environment. And we see this with PRRS and with a PCV PRRS challenge now. We've validated that. Diluting energy will increase feed intake to achieve that same goal. And really, what we don't know, is it more important to is it the protein or is it the essential amino acids or synthetic amino acids? Which one's better? We're doing a current study for that to see whether it's beneficial. And by far, this is the best, if we're talking about supporting pig growth during a health challenge from a nutrition standpoint, just changing the macronutrient composition, i.e. changing the lysine or amino acid to energy ratios, seems to be the best and most reliable way to actually improve growth performance during the health challenge period with PRRS. We do not know what that looks like with bacterial challenges, and that needs to be done with we need to look at that with bacteria to see whether we can achieve the same thing. But during a PRRS virus challenge, we're validated now on 4 reps of pigs, and we see the same thing, that we can improve growth rates. So really, in general conclusions, nutrient and partitioning and metabolism priorities are altered. In pathogen challenges, there is some changes in intestinal integrity due to PRRS, and also disease anorexia seems to explain a lot of the phenotype. All these disease challenges have a negative impact on growth and lean tissue accretion, and most of it seems to be due to nutrient supply, in other words, essential amino acid energy utilization or uptake by the pig, and feed intake, rather than actually repartitioning from muscle towards liver and other things. We see an increased energy demand due to PRRS. It's about 150 kcal of ME intake per day. The other thing is we've tried stuff that we can improve survivability but not have an impact on growth performance, and some other stuff has improved growth performance but didn't do anything with survivability. From what we're seeing right now, we've had all the nutritional studies that we've done, we've had a minimal impact or limited impact on the virology and serology aspects. In other words, on antibody kind of titers and CT values of PERS. We don't see nutrition really moving those numbers that much, and they hasn't translated into improved performance. But there could be some other things out there that may be improving or decreasing shedding time, and therefore that will then translate into, or improving seroconversion, that will then translate into improved performance from a nutrition standpoint. And the biggest thing is feed intake seems to describe most of the disease phenotypes that we've looked at. And so really, if we look in the future, what can we do to actually improve appetite or stimulate appetite without having any negative consequences on pigs? And then increasing amino acid density relative to energy seems to be a good practical solution that we've now tested 3 or 4 times, and we're improving growth rates and performance parameters in PERS challenge pigs. And so just to wrap up, to keep us within the hour, just want to acknowledge the Pork Board and the USDA for funding, and also the MASH-offs for helping us do the last 2 projects with lysine energy ratio work. Without the pork checkoff and also the MASH-offs, we wouldn't be able to do this kind of practical work. And then I'd like to thank Wes Schwier, Shelby Curry, Emma Helm, Carson DeMille, Blair Todd, and Jessica Jasper for doing a lot of the work that we've presented here. And with that, I'll be happy to take any questions, Chris.
Okay. Thank you, Nick. There was quite a few questions that came in during the question and answer period that were sent in by the chat box. Just to remind those of you that are listening in, if you want to type an additional question, please do so, and then I'll relay it to Nick. So the first question here, Nick, is if we can drive appetite, we can recover performance. And then if that's so, what are your recommendations for getting sick pigs to eat?
Well, I think from a practical standpoint, right now there's not a lot we can do during that peak infection period to get pigs to eat. And so I think there needs to be some more basic research to see, okay, what can we actually do to drive appetite during peak infection? I think the hypothalamus, where the appetite regulation is controlled, is overriding the rest of the body because it's probably not advantageous to drive feed intake during a pathogen challenge for 2 reasons. One, they probably think the pathogen is coming from the environment, so why bring more of it into the body? And secondly, they don't want to support any more kind of thermal effect of eating, so therefore they don't want to increase the fever response just through digestion, because that could be detrimental as well. So we don't know enough basic information about if we can actually drive it. We're doing some stuff in the next, hopefully, 6 to 12 months to actually see what's going on in the brain, to see if we can drive appetite during those health challenges. But from a current standing, what we do see in a lot of our disease challenges is that there is some compensatory gain that happens once they've seroconverted or they've stopped shedding the bacterial virus. And so I think there's opportunity to try and, whether it's increasing essential amino acid requirements or even vitamin mineral requirements, post-challenge, to actually, are we optimizing compensatory gain aspects in certain genotypes or certain genetics? We may not be able to do too much in maternal line genetics, but I think in some of the terminals, I think there's a lot more opportunity there to play on compensatory growth. And I think that's where we can probably drive more feed intake. So like I highlighted with the influenza nursery study that we got data from NutriQuest and Dr. Geiger at the vet school, those pigs wanted to eat more once they kind of got over the flu. And so can we do something? Feed intake's already up because they're trying to compensate. So are we actually feeding them the right requirements to optimize growth recovery? that's
Yeah,
going to be
that
cost
happened
effective.
relatively quickly. I was shocked by how fast that they started to eat more after the flu challenge.
About 7 to 14 days after the challenge. So it was only about a week or two. And we see that with, if you look at PD in nursery grower pigs, it normally just affects them for 2 or 3 days. They have diarrhea, and then they're kind of over it. So it happens relatively quick. But then, is there something that we can do on the back end to actually optimally support growth?
Mm-hmm. So that kind of goes along with this question that came in. In your model, when were the diets started relative to when the health challenge occurred? So I think what they're saying is, were they sort of front-loading? Were there any of your diets that were front-loaded?
So the diet stuff that we did in this current work, so with the PERS work that we did with Lysine to Energy, we actually started at the day we inoculated the pig.
Okay.
So the DPI 0, we started it right there. Now there's some practical. Practically, you probably would start it about a week later, because it takes you probably, once you figure out, there's circulating virus in the barn, the disease is showing clinical signs, you've got your diagnostics back, you're talking about probably 7 to 10 days. From a practical standpoint, that's where you may come in to actually say, well, what can we do? But we started at DPI 0, so day post-infection 0, or post-inoculation 0, because we just want to know, is the requirement changed there, and then also, is it beneficial? Another thing is, if pigs are coming in with a virus and we switch them onto it, we know they're coming in with a pathogen burden or a PERS load. Can we put them on this diet, or give them a diet straight away? And then, so that was our rationale at the time. But it's timing, severity is everything with all this stuff. And then it's horses for courses, because there's so many different combinations.
Yeah. So, question came in about, has the gut microbiome been investigated in these challenge models that you use?
On the PERS pair-fed study, I gave Tom Burke. I gave some isolated genomic material to Tom Burke at Nebraska, and he's actually looking at some of the microbiome, doing some microbiome analysis on those extracted fecal samples or luminal content samples. And so that's the closest that we've done with the microbiome side of things. I think Kansas State's done a lot more at least in the last year or so, with regard to microbiome and disease. But we haven't done a lot with the microbiome and disease. We are moving with the dietary fiber and our brachyspire, or the swine dysentery study. We are going to explore some of that stuff. I think with bacterial challenges, there's more opportunity with the microbiome than necessarily a virus challenge, where I sit and what I've seen so far. But there is an interaction. There is an interaction based off the Kansas State data. There is an interaction between PERS and the microbiome, but in the field, it's kind of messy.
Yeah. Yeah, I think the feeling, or the question was based on the idea that perhaps the microbiome would explain some of the differences in dietary treatments that you have applied. So, if particularly with fiber in the diet, that might indicate a return to a more normal microbiome.
Yes, it's either a return to a more normal microbiome, but whatever normal is, or it's actually actually the microbiome that's been promoted is actually been able to have some anti-pathogenic properties. In other words, they just outcompete swine dysentery or the brachyspire, outcompete Lawsonia, not allowing it to colonize and actually then come in contact with the epithelium. So that could be another way, or it could be promoting mucus production and then kind of creating a better barrier or absorptive barrier, secretory barrier. So
there's a lot we don't know there yet.
Correct.
So this one's going to switch gears just a little bit. Do you anticipate the same results with increased soybean meal with health-challenged young pigs as you observed in the ones with late finisher?
I think based off the Illinois data, so from what Dr. Dilger's published, depends on the parameter we're talking. If we're talking about improving growth performance with high soybean meal, they went up to about 40% soybean meal, and then over a very short duration, but they didn't really move the needle much with regard to growth rates in those pigs. They had improvements in some kind of virology, serology, and some blood data, but didn't translate to growth rates. So I think in a younger pig, I think there's some potential benefits, but there needs to be a bit more of a longitudinal study than just 14 to 21 days that the Illinois research has shown. But then what we're seeing is it seems to be you have to go over 40% soybean meal to see that benefit if we're talking about purely just soybean meal itself in a younger pig. Isoflavones may play a role in it, but you got to consume a lot of soybean meal to really get enough isoflavones into it, into the pig to actually have a beneficial effect. Dr. Laura Greiner did her PhD work looking at isoflavones, and what she showed benefits, but to actually get that much into the pig, she's isolated isoflavones. I mean, you have to consume probably 50-60% of the diet would have to be soybean meal to get that amount into the pig, which is not practical. Not practical,
yeah. All right, this is the last question I've got then, Nick, and then we'll conclude. But by increasing the lysine to metabolizable energy up to 110 to 120%, you mentioned improved performance compared to non-challenged pigs. Was this significant, statistically significant? Was that finding significant?
We didn't. No, it was compared to it wasn't compared to non-challenged pigs. It was compared to a 100% pig.
Okay. Yep.
So we did not statistically compare a healthy pig versus a PRRS challenged pig because they were in two different barns. We didn't do that, but when we did breakpoint analyses. So the first Schweer paper in Translational Animal Science in 2017 or 18 that we've published, what we see there is that actually the control pigs or the healthy pigs were breaking closer to where 100% should be on the breakpoint analysis, where the actual PRRS challenge pigs were actually breaking more around 110, 120%. So that's where the break points in those curves were showing, whether it's linear or quadratic analysis.
Yeah.
And then in the follow-up study, all we did was just compare everything was challenged, and then we just had 100%, 120% driven by increased amino acids and protein, or 120% by diluting energy with sand. And so we don't have a true control for those ones, like a negative control.
Right. Okay, well, we're past the top of the hour now, folks, so if there's no additional questions, I'd like to thank Dr. Gabler for an excellent presentation of the results of his research projects and that area of focus that he's had for perhaps the last 8 years, funded by checkoff dollars. I'd also like to remind our listeners that we will have one more webinar in this series. Next week, Dr. Jason Ross will share an overview of the effort that he has been coordinating to improve pig survivability. In his effort, this is a consortium of 17 researchers from 3 different institutions who will dedicate substantial expertise and focus to this area over the next 5 years. If you've not yet registered for that webinar, please remember to do so at pork.org. I hope you found the information presented today to be useful in improving the profitability and performance of your enterprise. I look forward to you joining next week's webinar. Thank you all very much and have a great day.
Yep. Thank you, everyone.
Thanks, Nick.
All right.